Hello everyone, welcome to Get My Class. So let's solve this question which says nematode-specific genes were introduced into the host cell by using option A Salmonella vectors, option B E. coli vectors, option C Agrobacterium vectors, and option C Bacillus thuringiensis. Now let us understand the question. It says nematode-specific genes were introduced into the host plant by using what? Now when we say nematode-specific gene, why are we talking about nematode-specific gene? What are these nematodes? These nematodes are type of disease causing organism, okay, which cause disease in tobacco plants. Okay, basically tobacco. These are type of parasites. Okay. Now these nematodes, they produce certain proteins which will cause disease. Okay, this will cause what? This will cause disease in these plants and hence reduced yield. Reduced yield. So what we have done to block this production of proteins, we have to block the mRNA because we know that DNA of the nematode will give rise to mRNA. That is the central dogma, right? mRNA, and further mRNA will give rise to proteins, and that will cause the disease. So what we are trying to do, we don't want this protein to be produced. So we are going to block this mRNA so as to block the proteins. How are we doing? We are actually isolating the specific gene from nematode, that is nematode specific gene, and we are introducing into the vector. Okay, vector. Which vector? The Ti vector or Ti plasmid. And Ti plasmid belongs to what? Agrobacterium tumefacium. Okay, and this, of course, it's a DNA vector. Okay, because it is all vectors are DNA vectors. So here we are taking specific gene. The DNA, we know that gene when we say it, we are talking about the DNA. Okay, the gene which is present, it is a part of the DNA. Now that gene will be inserted into the Ti plasmid. This will code for. So once this is introduced, there is formation of the sense RNA and antisense RNA. So this whatever the complementary strands are there, which are produced by the gene from the plasmid, that will go and form double-stranded structure along with the mRNA of the insect itself. So then what will happen? It will become double-stranded, hence no protein formation because double-stranded when it forms, it will not translate. It will not get translated. Hence, there is no production. So that is the entire technique. So let us see. So nematode-specific genes were introduced into the host by using Salmonella vectors. No. What are Salmonella vectors used for? Of course, these Salmonella vectors are used for cloning specific type of genes. Okay, but this is not used to infect the whole. I mean, it is not used to transfer the DNA into the host cell here with respect to the tobacco plant. Okay, hence it is ruled out. Option B says E. coli vectors. So E. coli vectors are used for many cases. Okay, for production of human insulin, etc. But in this case of nematode specific genes, these were not used. Option C says Agrobacterium vectors. Yes. So as I told you, the Ti plasmid which belong to Agrobacterium. Hence that is the correct answer. Option D says Bacillus thuringiensis. This is used for the production of Bt toxin and not for antisense RNA in case of these particular type of tobacco plant. Okay. Hence the correct answer is what? Option C. So let us conclude. What is the correct answer? Option C. I hope you understood the explanation. Thank you.